The overall goal of this study is to compare two different methods of human monocyte isolation for dendritic cell generation in vitro and to characterize the CD11C, CD14 cell surface expression of the resulting monocyte-derived populations by imaging flow cytometry.
These monocytes isolation protocols can contribute to the development of reliable methods for the purification of human dendritic cells for research and clinical applications.
The main advantage of these protocols is that they facilitate the isolation of human monocytes and their differentiation into viable and functional monocyte-derived cells. In addition, this is the first study to characterize a specific population of monocyte-derived dendritic cells by single-cell imaging flow cytometry.
This method can also be applied as an alternative for the isolation of other rare cells at an adequate yield for downstream research applications.
Visual demonstration of these methods is critical, as it can be challenging to handle the blood sample safely without the appropriate instruction and experience.
Begin by diluting the blood sample to a 1:1 ratio with PBS in a T75 flask. Then, add 15 milliliters of density gradient solution into a 50 milliliter centrifuge tube and carefully layer 25 to 30 milliliters of the diluted blood over the gradient.
To achieve a proper separation of the peripheral blood mononuclear cells, load the blood onto the density gradient solution quickly but carefully and without mixing the layers.
Separate the cells by centrifugation, followed by the transfer of the white blood cell interface layer into a new 50 milliliter conical tube.